So, welcome everyone. Welcome to this series of general physiology. Okay, so we are in the end section of this chapter now. So, we are going to see the gene. Okay, so let's start the gene in this lecture. Okay, so we have discussed in the previous lecture that the gene is a portion of the DNA. Okay, so DNA is a double stranded structure. Okay, like that. In this double stranded structure, there are some portion which carry some specific hereditary information that some part of the strand is called a gene, okay, which carry a specific hereditary information, okay. So that's basically gene which carry message or code for synthesis of specific protein, okay, from amino acid. It is like a book that contains the information necessary for the protein synthesis. Gene is considered as a basic hereditary unit of a cell. It is a basic hereditary unit of a cell. So when DNA wrap around itself, it forms the chromosome. Okay. So chromosome is a unit of DNA and gene is a unit of DNA is a DNA is a unit of chromosome and gene is a unit of DNA. So gene there is a formation of DNA. Okay, and from DNA there is a formation of chromosome. So basically gene is a basic hereditary unit of a cell. Okay. Gene is a segment of DNA which carries specific information for the formation of specific protein. Okay, and so by many gene DNA is a form, and by many DNA there is a formation of chromosome. Okay, in the nucleotide of the DNA there are the successive base pair of together called as triplet. So the strand of DNA is made up. In the strand of DNA, there is a presence of some nitrogenous bases, which include adenine, guanine, cytosine, and thymine. Adenine, guanine, cytosine, and thymine. Okay, in this nitrogenous base, three successive base pairs are together called as triplet or codon. These three pairs are called as triplet or codon. Each codon codes for a code word information for one amino acid. Okay, one amino acid. There are 20 amino acid, and there is a separate code for each amino acid. For example, the triplet CCE code for the glycine and GGC code for the protein. Okay, so that is what it is. Thus, the gene forms the code word for a particular protein and to be synthesized in the ribosome outside the nucleus from amino acid. So, gene is basically a segment of DNA which carry information for the formation of protein, protein is synthesized outside the nucleus in the ribosome from amino acid. Okay, and the three successive codons in the strands of DNA. These three successive codons is known as triplet. Three successive base pairs are known as triplet, and also known as a codon, which code for the synthesis of a particular amino acid. So there are 20 amino acid, and from that 20 amino acid, there is a formation of proteins occurs. Okay, so that's what it is. So genetic disorder. Genetic disorder, it is occurred due to the abnormality in the individual genetic material. Okay, genetic material may abnormality. So genetic disorders are either hereditary disorders or due to the defects of gene. So cause of genetic disorder, genetic variation and genetic mutation. So presence of different form of a gene. Okay, presence of a different form of a gene is called a genetic variation. Means in the DNA some different gene is present. Okay, instead of normal gene some defective gene is present. Okay, some different gene is present. And genetic mutation, generally the mutation means alteration or change in the nature, form or quality of a genetic mutation refers to the change of the DNA sequence within the gene or a chromosome of an organism which results in the creation of the new character. Okay, so alteration of sequence or change in the sequence of a base pairs of a gene is known as genetic mutation. Mutation is basically meaning change. The meaning of mutation is basically change. Okay, and genetic variation is a present of different form of a gene. When the entire gene is different, okay, then we call it as a genetic variation. Okay, but in genetic mutation, the sequence of the base pair is changed. Okay, the sequence of a base pair is changed. Okay, which result in the creation of the new character. So that's why that is called a genetic mutation. So classification of genetic disorder: single gene disorder, multifactorial genetic disorder, chromosomal disorder, and mitochondrial DNA disorder. mitochondrial DNA disorder, single gene disorder, chromosomal disorder, and multifactorial genetic disorder. Single gene disorder, multifactorial genetic disorder, chromosomal disorder, and mitochondrial DNA disorder. So first one is a single gene disorder. Single gene disorder or the Mendelian or the monogenic disorder occur because of the variation or the mutation in the one single gene. When there is a variation or the mutation, when there is a variation or the variation means completely different gene, mutation means change in the or the alternation of sequence of the base pair in the particular gene. That's called mutation. Okay, in single gene, that we call it the single gene disorder. Examples of the single gene disorders are the sickle cell anemia and the Huntington's disease. Okay, so multifactorial genetic disorders include or the polygenic disorder. Poly means many. Okay, poly means many. Means here uh, there is a variation and mutation in multiple genes. Okay, it is occur by the combination of environmental factor and mutation in the multiple gene. Variation and mutation in multiple gene. So some environmental factor also responsible for this multifactorial genetic disorder. Okay. Okay. So examples of these multifactorial genetic disorders are the coronary heart disease, Alzheimer's disease, arthritis, and diabetes. Coronary heart disease, Alzheimer's disease, arthritis, and diabetes. Okay. Third one is a chromosomal disorder. So chromosomal disorder are due to the abnormality in entire chromosome. Abnormality in entire chromosome. Okay. It also called as chromosomal abnormality, anomaly or aberration. It often results in genetic disorder which involve the physical or the mental abnormality. So chromosomal disorder is caused by the numerical abnormality or structural abnormality. Either there is a change in the number of chromosome or either there is a change in the structure of chromosome. There is a two types of abnormality in the chromosome. That is the numerical abnormality and the structural abnormality. Okay. So structural abnormality means called as alternation. Okay. Uh, Alternation of chromosome which lead to disorders like chromosomal instability syndrome. Okay, chromosome instability syndrome. So numerical abnormality, it is of two types. First one is a monosomy due to absence of one chromosome from a diploid number. So there are 23 pair of chromosome. 23 pair means there are 46 chromosome. So there is absence of one chromosome. That is means there are only 45 chromosome. So examples is a Turner syndrome. Okay, which is characterized by the physical disability. Okay, trisomy due to the presence of one extra chromosome means there are 47 chromosome. Okay, so example is a Down syndrome. Example is a Down syndrome. Characterized by the physical disability and mental retardation. So in numerical different deformities of two types, monosomy and trisomy. In monosomy there is absence of one chromosome, so hence there are 45 chromosome. Examples of the monosomy is Turner syndrome. Turner syndrome. And trisomy there are present of one extra chromosome, hence there are 47 chromosome instead of 46 chromosome. So examples of the trisomy is a Down syndrome. Okay, so mitochondrial DNA disorder. Next one is the mitochondrial DNA disorder. Mitochondrial DNA are the genetic disorder caused by the mutation in the DNA of mitochondria. So we have discussed in the previous lecture that the mitochondria has its own DNA. So extra DNA is present in the mitochondria outside the nucleus. Okay, so when there is an alternation in this DNA of the mitochondria, that is a non-chromosomal DNA. Okay, this DNA does not form chromosome. Okay, that is a non-chromosomal DNA. Okay, so some diseases are occur. These diseases are Kearns-Sayre syndrome. Kearns-Sayre syndrome. It is a neuromuscular disorder characterized by the myopathy, cardiomyopathy, and paralysis of ocular muscles. Okay, myopathy, cardiomyopathy, or paralysis of the ocular muscle, Kearns-Sayre syndrome, and Leber's hereditary optic neuropathy. Leber's hereditary optic neuropathy. This is characterized by degeneration of retina and loss of vision. Okay, so there are two disorder caused by the mitochondrial DNA disorder. First one is a Kearns-Sayre syndrome. First one is a Kearns-Sayre syndrome, and second one is a Leber's hereditary optic neuropathy. Kearns-Sayre syndrome and Leber's hereditary optic neuropathy. So this is a double-stranded structure of the DNA. This is a strand of the DNA. This strand is made up of sugar and phosphate. This is the backbone of DNA. Sugar present in this DNA is deoxyribose sugar. Okay. strand, there is a presence of this nitrogenous basis. Okay, this nitrogenous basis. Adenine pair with thymine, cytosine pair with guanine, 2 hydrogen bond, and by 3 hydrogen bond. So I think you all have studied it in the 12th class. Okay, so there is nothing much to explain about it. We will move forward. Next one is RNA. Next one is RNA. This is all about DNA. So RNA is a nucleic acid that contains the long chain of nucleotide unit. Okay. So there are some difference between the DNA and RNA. It's basically simple because RNA is a single stranded, unlike DNA. Okay, DNA is a double stranded, RNA is a single stranded. Okay, but in RNA there is presence of ribose sugar, and DNA there is a presence of deoxyribose sugar. So the sugar present in RNA is ribose instead of deoxyribose. Okay, and there is a difference in the nitrogenous bases also. So instead of thymine in the DNA, there is a presence of uracil in RNA. Okay, and so various functions coded in the genes are carried out in the cytoplasm by the cytoplasm of the cell by RNA. So we have discussed that the gene carry information for the synthesis of protein. For that protein synthesis, RNA help. RNA carried out many functions in the process of that protein synthesis. Okay, so basically RNA is formed from DNA. Okay, RNA is formed from DNA. So we have discussed this earlier. So RNA is of three types: messenger RNA, transfer RNA, and ribosomal RNA. Okay, each type of RNA is play a specific role in the protein synthesis. Okay, so basically main functions of the RNA is the protein synthesis. The synthesis of protein from the information of the gene. Okay, from the DNA. So these are three types of the RNA. In messenger RNA, carry genetic code of amino acid sequence for synthesis of protein from DNA to cytoplasm. So that messenger RNA carries a message for the synthesis of protein from DNA to cytoplasm. Okay. Okay. Amino acid sequence. Genetic code of amino acid sequence. Okay. So we have discussed that the particular codon, particular triplet, particular three pairs of the particular nitrogenous bases form information for the synthesis of particular amino acid. Okay. So this information is carried out from nucleus to the cytoplasm, because we have discussed that the DNA is present in the nucleus, and this information is carried out from nucleus to cytoplasm by messenger RNA. Okay, transfer RNA is responsible for decoding the genetic message present in the mRNA. Okay, it decodes the genetic message present in the mRNA, means it should do the actual work. Actual work is done by the transfer RNA. Okay. Ribosomal RNA is present within the ribosome and forms the part of the structure of ribosome. It is responsible for the assembly of protein from amino acid in the ribosome. Okay. So ribosomal RNA is present in the ribosome. It forms the protein from amino acid in the ribosome. Okay, that's what. So gene expression. What is gene expression? Gene expression is basically the message in the gene for the synthesis of protein. Okay, for the synthesis of protein. By that message, actual product, gene product, or document of instruction, RNA is used for the protein synthesis. Okay, so that is what we call a gene expression. So gene expression is a process by which the information or the code word encoded in the gene is converted into the functional gene product. That information is converted into the functional gene product or the document of instruction, which is called RNA, and is used for the protein synthesis. Okay, so basically in simple language, it is a actual work. It is actual action. Okay, on that message in the gene. Okay, so gene expression involve the two steps, transcription and translation. Okay, transcription means the synthesis of RNA from DNA. Okay, so basically in this process of gene expression, there are two main steps. First one is a transcription, second one is a translation. So in word transcription means copying. It indicates the copying of genetic code from DNA to RNA. So basically RNA is formed from DNA. So information present in the DNA for the synthesis of protein is copied to the RNA. So the protein are synthesis in the ribosome which are present in the cytoplasm. Okay, protein are synthesis in the ribosome and ribosome are present in the cytoplasm. However, the synthesis of different protein depend upon the information. Okay, code sequence of the codon. Okay, encoded in the gene of the DNA which is present in the nucleus. Okay, since DNA is a macromolecule, it cannot pass through the pores of the nuclear membrane. DNA is a big molecule. The size of DNA is large, so it cannot pass through the pores of the nuclear membrane and enter the cytoplasm. But the information from the DNA must be sent to the ribosome. So the information for the synthesis of protein in the DNA, present in the DNA, must be sent for the ribosome. So because of the macromolecule, because of the large structure of the DNA, because of the double-stranded structure of the DNA, this information present in the DNA is transferred to the ribosome, present in the cytoplasm outside the nucleus. So gene has to transcribe. Transcribe means copied. Okay, transcribe means copied into mRNA which is developed from the DNA. So DNA forms mRNA. The information for the synthesis of protein present in the DNA are copied to the mRNA, and this mRNA, which is a single stranded, which can easily pass through the nuclear pores. Okay. Okay, thus the first stage in the protein synthesis is transcription of genetic code, means copying of the genetic code from DNA to mRNA, which occurs within the nucleus. Okay, it's involve the formation of mRNA and simultaneous copying or transfer of information from DNA to mRNA. Okay, mRNA, which is a messenger RNA. Okay, the mRNA enters the cytoplasm from the nucleus through the nuclear pore, nuclear pore. Okay, and activate the ribosome, resulting in the protein synthesis. So formation of mRNA from DNA is facilitated by the enzyme RNA polymerase. Okay, this enzyme RNA polymerase. Okay, the enzyme responsible for this process, formation of mRNA from DNA, is occurred by the enzyme RNA polymerase. Second step in the gene expression is translation of the genetic code. Okay, translation of the genetic code. So translation is the process by which protein synthesis occur in the ribosome. So this is the actual process of synthesis of protein in the ribosome. So that's why I called it as a translation of the protein under the direction of genetic instruction carried by the mRNA from DNA. Okay, or it is the process by which mRNA is read by the ribosome or to produce a protein. This involves the role of other two types of RNA, namely tRNA and rRNA. Okay. mRNA moves out of the nucleus and cytoplasm. Now a group of ribosome called polysome gets attached to the mRNA. The sequence of codon in mRNA are exposed and recognized by the complementary sequence or the base in the tRNA. Okay, so we have discussed the functions of RNA, that the functions of tRNA is a decoding of the information. Okay, so it decode the information, it gain the information from mRNA. So the base pair present in the mRNA, the tRNA form the complementary sequence of the base. Complementary base are formed. The complementary sequence of the base is called as anticodon. According to the sequence of the base in the anticodon, different amino acids are transported from the cytoplasm into the ribosome. So by tRNA that act as a carrier. Okay, with the help of RNA, protein synthesis are assembled from the amino acid. Okay, the protein synthesis occur in the ribosome, which are attached to the rough endoplasmic reticulum. So it is synthesis of with the help of the anticodon, amino acid are bring to the ribosome. Okay, so with the help of that amino acid, protein are synthesized in the ribosome, which is attached to the rough endoplasmic reticulum. Okay, which is attached to the outer surface of rough endoplasmic reticulum, and rough endoplasmic reticulum is continuous with the nuclear membrane. Okay, so that is what it is. So this is all about the synthesis of protein, how protein are synthesized. So that is what transcription and translation. Okay, thank you. So please share suggestions or thought in the comment section. Let me know any improvement you want in the teaching method. Okay, thank you.